So this is our key. There's a nucleolus, which is the blue dot. Chromosomes, which is like the rectangles that are attached together. The sister chromatids are like the waves, and the cell membrane is just the area around the entirety of the image. The T is a centriole. The orange dots are DNA, and the blue circle is the nuclear envelope, and the orange fibers are spindle fibers. So in G1, also known as synthesis, we start off with a very, very small cell, and as time moves on, it gets bigger and bigger, and also DNA and chromatids are also copied as the cell gets larger. So,
of interphase, the nuclear envelope encloses the nucleolus, which is the small blue dot there. The nucleus contains one or more nuclei. The two centrosomes, which are the T's, have formed by duplicating by a single centrosome. And chromosomes have been duplicated by the S phase, and you can see them, they're all in there. And they are duplicated and uncondensed. And that is G2 of interphase. So this is prophase. The nucleoli has disappeared, as you can see. Each duplicated chromosome appears to have an identical sister, as you can now see, called chromatids. The meiotic spindle has started, the spindle fibers between the two centrosomes containing the centrioles. And the centrosomes have begun to move away from each other. That is prophase. So this is prometaphase. The nuclear membrane has begun disappearing, hence the dotted lines. The centrosomes containing the centrioles have moved very far away, so therefore the spindle fibers have been stretched immensely. And you can see that the chromosomes have started forming, hence the rectangles, and that is prometaphase. Metaphase, the chromosomes have completely formed, and those are the rectangles going across the cell, and the centrioles and the centrosomes containing the centrioles are still at the complete opposite ends of the cell, and the spindle fibers are still linking them, and that is metaphase. Anaphase is the shortest stage of mitosis, and it only lasts a few minutes. The sister chromatids are starting to break apart, and the spindle fibers are pulling them towards the centrioles. As you can see, the anaphase is the cell is slightly bigger as it's in the process of breaking apart, and you can see only this middle chromosome is keeping this thing together, and the centrioles are still at opposite ends, containing the spindle fibers that are attached to the sister chromatids. This is telophase. The cell is almost broken apart completely. The chromosomes, the sister chromatids, have been redone in their nuclear envelopes, and then once these two break apart, it goes back into this cell, the start of G1, and that is the cell cycle. So during the G1 and synthesis phase, cyclin then accumulates all the way through, and then MPF at the after G2 of interphase, the accumulation of cyclin results in the MPF pushing it towards the mitosis phase, and that results in prophase through telekinesis.